Genebass was designed as a multi-phenotype association browser. To see other LDLR associations, we can change the results view to the gene PheWAS view. This brings up a tool for browsing all LDLR gene associations in the study, which comes out to just under 3,700 phenotypes. Expand the panel to see more columns. There's also a set of controls for searching or filtering phenotypes and changing what data is displayed. There are two plots, one for showing association p-values, where each point represents a phenotype colored by its category. There's also a smaller plot for showing betas for each phenotype. Using these buttons, I can choose to show just p-values, just betas, or both. The table shows phenotypes ordered by p-value. Sometimes the plot can be dominated by very strong hits, and you may want to see hits closer to the significance threshold. You can choose to plot p-values on a double log plot or tweak the cutoffs using the sliders. I can control the width by dragging the center of the page or by pressing the shortcuts at the top. You can choose the full screen mode to get the full page view. This allows you to see more columns. Or you can choose to hide this page entirely. We can also show or hide control panels on both sides to make more room for plots and tables. It's possible to filter the phenotypes by keyword, for example, searching for cholesterol. We can also browse the UK Biobank showcase categories. Clicking on a category will filter the phenotypes down to those belonging to that category. We can display p-values ordered within each category as well. Note the p-value legend showing the thresholds and coloring scheme of the circular p-value indicators. Betas are also colored by direction and magnitude. More blue for positive, more red for negative. Clicking on these black arrows will update the gene page to display results for that phenotype on the right-hand side. The active phenotype is circled in blue on the plot, and also indicated by a little checkbox in the table that is filled. We can also click directly on the PheWAS plot to update the phenotype. Remember, there are 3 functional annotation categories and region-based statistical tests for a total of 9 p-values per gene. The p-value column currently displays scat-O results for the p-LOF class. The active annotation class is always displayed at the top of the screen. So currently I'm looking at the p-LOF scat-O p-values across all phenotypes for LDLR. I can change this to missense and look at the p-values coming from burden tests, and now I'm effectively looking at this cell in the table across all phenotypes. Note that only the burden test will produce gene burden betas, and these are always displayed, regardless of which region-based test is selected. If you have scat or scat-O selected, you'll get this warning at the bottom indicating that these values are coming from different tests. There's also a notice saying some phenotypes are hidden. This can happen if you filtered phenotypes using the controls. In this case, it's because we don't have the include filtered checkbox checked, and this is the default state. Clicking this checkbox will include phenotypes that failed our QC processes, so use this with caution. After clicking it, we're no longer hiding phenotypes, but we notice that we've added some that have indicators with faded colors and dashed lines. Inspecting this more closely, we see that this phenotype has failed the lambda GC cutoff. Phenotypes not indicated by green or orange will take a bit longer to load. This is because they're not cached by the server, so it takes a bit more work to retrieve this data. Finally, we can export the current set of phenotypes to CSV.